According to the last classes, in eukaryotic cell, either plant cell or maybe animal cell. Today for this class, I took an example of one of the eukaryotic cell, plant cell. The size of plant cell is bigger than the animal cell. From the last class, we discussed the structure of plasma membrane and their chemical composition we discussed, and the cell wall and their chemical composition in different organisms also is completed. Now, interior to cell or inside the cell, there is a dense matrix substances is present, is called as cytoplasm. Cytoplasm is the main arena for all biological activities. Clear? Then next, these cytoplasm having different membrane bound cell organelles, either double membrane bound cell organelles or single membrane bound cell organelles. Even membraneless cell organelles also is present in eukaryotic cell. So here the nucleus is a double membrane bound cell organelle. It has a distinct structure and a distinct function. And mitochondria is a double membrane bound cell organelle having distinct structure and a distinct function. And chloroplast also having double membrane bound cell organelle having distinct structure and distinct function. And endoplasmic reticulum having single membrane bound cell organelle, and Golgi complex having single membrane bound cell organelle, and lysosomes having single membrane bound cell organelles, and vacuoles also having single membrane bound cell organelles. Even peroxisomes also single membrane bound cell organelle. Along with it has even membraneless cell organelles. These membraneless cell organelles we called as ribosomes. So each and every cell organelle having distinct structure and distinct function in the cytoplasm of eukaryotic cell. Out of all the cell organelles, a few cell organelles functions coordinate to each other. Such kind of cell organelles we called as endomembrane cell organelle. Such kind of system is called as endomembrane system. Endomembrane system is absolutely absent in prokaryotic cell. Endomembrane system we are seen in eukaryotic cell, such as endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. These four cell organelles we called as endomembrane cell organelles. Region is from endoplasmic reticulum, Golgi complex is formed. From Golgi complex, lysosomes is formed. From lysosomes, vacuoles are formed. So that's why these four cell organelles, their structural and functional aspects coordinate to each other. So that's why these cell organelles is called as endomembrane cell organelle. This system is called as endomembrane system. We are seen in eukaryotes, absent in prokaryotes. Region is the endoplasmic reticulum coming from nuclear membrane. In prokaryotes, nuclear membrane is absent. And here the chloroplast cell organelle, mitochondria cell organelle, ribosomes and peroxisomes are present. But these cell organelle function is not coordinated. So that's why these are not comes under endomembrane cell organelle or endomembrane system. So today in this class we discuss endomembrane system. In endomembrane system, how many cell organelles are present, Amma? 4. Endoplasmic reticulum, Golgi complex, lysosomes, vacuoles. Let's we discuss endomembrane system. In endomembrane system we are seen in eukaryotes. In endomembrane system, 4 cell organelles are present. These 4 cell organelles are coordinated to each other for their structures and functions. The first one is endoplasmic reticulum. The second one is Golgi complex or Golgi apparatus. And the third one is lysosomes. Lysosomes. And the fourth one is vacuoles. So in this class we discuss these four endomembrane cell organelles or endomembrane system. All these endomembrane cell organelles are single membrane bound cell organelles. Clear? Single membrane bound cell organelles. A single membrane bound cell organelles. Single membrane bound cell organelles. All endomembrane cell organelles are single endomembrane bound cell organelles. Here the first endomembrane system is endoplasmic reticulum. Let's we discuss endoplasmic reticulum. Simply we called as ER. Endoplasmic reticulum. Endoplasmic reticulum simply we called as ER, which is the first endomembrane system cell organelle amma? ER. ER stands for endoplasmic reticulum. For example, this endoplasmic reticulum was first discovered by K.R. Porter. Who was the scientist first discovered endoplasmic reticulum? Means K.R. Porter. The scientist K.R. Porter, he was first reported and first discovered endoplasmic reticulum in eukaryotic cells, either plant cells or animal cells. Now, this is single membrane bound cell organelle. Already I discussed with you, all endomembrane bound cell organelles are single membrane bound cell organelles. For example, if you can imagine this is an eukaryotic cell, listen carefully. For easy to understand, I would like to explain with eukaryotic cell diagrammatic presentation. For example, you can imagine this compartment is nothing but one eukaryotic cell, because endoplasmic reticulum or endomembrane system we are seen in eukaryotes, not seen in prokaryotes. Here, this is the nucleus. Clear? This is the nucleus. Clear, amma? This is nuclear is double membrane bound. Nucleus is double membrane bound. The outer nuclear membrane, it forms a network like structure. Outer nuclear membrane. This is inner nuclear membrane and this is outer nuclear membrane. This is the nucleus. Inside the nucleus, a subspherical like structure is called nucleolus and a network like structure is called chromatin material. This is the basic structure of nucleus and this is called as outer nuclear membrane and this is called as inner nuclear membrane. The outer nuclear membrane forms a network of tiny tubular like structures. Listen carefully, amma. A network like tiny tubular like extensions from outer nuclear membrane to plasma membrane. This is the plasma membrane of cell. This is the plasma membrane of cell. Once again, in eukaryotes, from outer nuclear membrane of nucleus, a small tiny network of reticulum means network. A network of reticulum of tiny tubular like extensions, tube like extensions from outer nuclear membrane to plasma membrane is called as endoplasmic reticulum. This endoplasmic reticulum was first reported or discovered by K.R. Porter. It is single membrane bound cell organelles. These endoplasmic reticulum generally made up of three different types of components. Endoplasmic reticulum is made up of three different types of components. The first one is cisternae. Cisternae is the first structure made to form endoplasmic reticulum. Cisternae means these are flattened sac-like structures arranged parallel to each other. The flattened sac structure arranged parallel to each other is called as cisternae. And the second structure is tubules. Tubules are either branched or unbranched structures is called as tubules. And the third one is vesicles. Vesicles are looks like a oval shape, spherical shape. These oval shaped vesicles and branched or unbranched tubules, a flattened sac-like structure cisternae, these three components made to form endoplasmic reticulum. So, these endoplasmic reticulum, based on their compartments, it is of two types. Inside the endoplasmic reticulum, there is a space is present. See, this is called as intraluminal space. And outside the endoplasmic reticulum, there is a space is present is called as extraluminal space. Clear for everyone? Now this endoplasmic reticulum is classified into two types. The first one is a rough endoplasmic reticulum. Which is the first type of endoplasmic reticulum? A rough endoplasmic reticulum. Simply we called as RER. RER means rough endoplasmic reticulum. If the surface of endoplasmic reticulum having ribosomes, if you touch this endoplasmic reticulum, it looks like a rough in nature because of ribosomes are present. Such kind of endoplasmic reticulum is called as rough endoplasmic reticulum. The second type of endoplasmic reticulum is SER means smooth endoplasmic reticulum. Reason is the surface of this endoplasmic reticulum not having ribosomes. So, based on the presence or absence of ribosomes, endoplasmic reticulum classified into two types. Rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is also called as granular endoplasmic reticulum. Regions, a granule like ribosomes are present on the surface of this rough endoplasmic reticulum. The smooth endoplasmic reticulum surface is not having any ribosomes, then it is called as agranular endoplasmic reticulum. Agranular means ribosomes are absent. So, based on the presence or absence of ribosomes on the surface of endoplasmic reticulum is of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Here the surface of this endoplasmic reticulum, if in case having 80S type of ribosomes, because the cytoplasm of eukaryotic cell having 80S type of ribosome, then the surface of these endoplasmic reticulum binded by 80S type of ribosome. For binding of these ribosomes on the surface of these endoplasmic reticulum, it required a gum-like structures. Those gums we called as glycoproteins. Glycoproteins such as riboforin 1 and riboforin 2. Riboforin 1 and riboforin 2, these are the two different types of glycoproteins responsible for attachment of large size of ATS ribosomes on the surface of endoplasmic reticulum. Now this endoplasmic reticulum we called as rough endoplasmic reticulum. So what is the function of rough endoplasmic reticulum, sir? Whenever ribosomes are present, endoplasmic reticulum main function is protein synthesis. Clear? So the main function of rough endoplasmic reticulum is protein synthesis. And these rough endoplasmic reticulum forms the second type of endoplasmic reticulum. Which is the second type of endoplasmic reticulum, amma? Smooth endoplasmic reticulum. The rough endoplasmic reticulum forms smooth endoplasmic reticulum. The outer membrane of nucleus produces rough endoplasmic reticulum, and rough endoplasmic reticulum forms smooth endoplasmic reticulum. The surface of this endoplasmic reticulum not binded by any ribosomes, then it is called SER, smooth endoplasmic reticulum, or agranular endoplasmic reticulum. The main function of these smooth endoplasmic reticulum is this function. Rough endoplasmic reticulum main function is protein synthesis, because the surface of this endoplasmic reticulum having ribosomes, and it forms smooth endoplasmic reticulum. Smooth endoplasmic reticulum means if the surface of endoplasmic reticulum not having any ribosomes is called as smooth endoplasmic reticulum. The main function of smooth endoplasmic reticulum, synthesis of lipids and steroids. Steroids means hormones, sex hormones. And one more, the smooth endoplasmic reticulum present in muscles, those smooth endoplasmic reticulum is called as sarcoplasmic reticulum. For example, this is my hand. In my hand many muscles are present. So the contraction and relaxation of my body and my muscles because of uptake and release of calcium ions by smooth endoplasmic reticulum in the muscle. What is the name of smooth endoplasmic reticulum in the muscle? Sarcoplasmic reticulum. The smooth endoplasmic reticulum present in the muscles is called as sarcoplasmic reticulum. This sarcoplasmic reticulum, whenever it's intake the calcium ions, then it is responsible for contraction. Whenever it releases calcium ions, it is help for relaxation. The contraction and relaxation of muscles due to uptake and release of calcium ions by one of the smooth endoplasmic reticulum in muscles is called as sarcoplasmic reticulum. And one more, there is no ribosomes, then the main function of these smooth endoplasmic reticulum is synthesis of lipids and steroid sex hormones. And one more, whenever we take any drugs, if the drug having toxic nature, means poison nature, such kind of toxic drugs is detoxified. Toxic nature will be removed from the drugs by the help of smooth endoplasmic reticulum. Means detoxification of drugs. The detoxification of drugs in our body by help of which of the following type of endoplasmic reticulum means smooth endoplasmic reticulum. For NEET competitive entrance exam, this point is very most important. And one more, the rough endoplasmic reticulum releases any components, either proteins or any substances. It is transfers to the next endomembrane cell organelle is Golgi complex, which is after endoplasmic reticulum, which is the next one, Golgi complex. The transfer of rough endoplasmic reticulum material to Golgi complex through the smooth endoplasmic reticulum. See here, for example, here see, This is smooth endoplasmic reticulum. The transfer of materials from rough endoplasmic reticulum, for example, rough endoplasmic reticulum main function is synthesis of proteins. Those proteins are packed in vesicles. These vesicles are transferred from rough endoplasmic reticulum to the next endomembrane cell organelle is Golgi complex via smooth endoplasmic reticulum. These are the information about endoplasmic reticulum. Once again, I recall headlines of first endomembrane system of cell organelle is endoplasmic reticulum. Simply we call ER. It was first discovered by K.R. Porter. Here. And it is a single membrane bound cell organelle. It is seen in eukaryotes. It is a endomembrane system cell organelle. It is mainly made up of three components: cisternae, tubules, vesicles. So, based on the formation of space, this endoplasmic reticulum forms two different space, intraluminal space and extraluminal space. Based on the presence or absence of ribosomes on the surface of endoplasmic reticulum, endoplasmic reticulum is of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Ribosomes present on surface of rough endoplasmic reticulum, ribosomes are absent in smooth endoplasmic reticulum. Rough endoplasmic reticulum main function is protein synthesis. Smooth endoplasmic reticulum main function is lipids and steroid synthesis, muscle contraction and detoxification of drugs. The transfer of components from rough endoplasmic reticulum to Golgi complex via smooth endoplasmic reticulum. A smooth endoplasmic reticulum present in the muscles is called sarcoplasmic reticulum. Clear? The next endomembrane cell organelle is Golgi complex. After endoplasmic reticulum, the second endomembrane cell organelle in eukaryotic cell is Golgi complex. Golgi complex. In the year of 1898, one of the dynamic scientists, his name is Camillo Golgi. The scientist Camillo Golgi, in the year of 1898, he was first reported and he was first discovered Golgi complex in eukaryotic cell. After his name, Camillo Golgi, this apparatus we called as Golgi apparatus or Golgi complex. See here. For example, this is nucleus. This is nucleolus, chromatin material, inner nuclear membrane and outer nuclear membrane. And outer nuclear membrane forms a tiny reticulum of network of tubular like structure is called endoplasmic reticulum. Based on the presence or absence of these ribosomes, endoplasmic reticulum is of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Already it's over from the last class. This endoplasmic reticulum main function is synthesis of proteins. This endoplasmic reticulum main function is synthesis of lipids and steroidal hormones. The next endomembrane cell organelle is Golgi complex. A network-like structure, a darkly stained, once again, a darkly stained, a darkly colorful network-like structure present very near to nucleus in eukaryotes is called as Golgi complex. It was first discovered by Camillo Golgi in the year of 1898. The structure Golgi complex is made up of four different types of components or four different types of structures. The first structure is called as cisternae. You know very well. Same component to form endoplasmic reticulum. These are flattened structure arranged parallel to each other. These are sac-like structure. Each Golgi complex is made up of 4 to 8 cisternae. This is important. And each cistern measures 0.5 millimicrons to 1 millimicrons diameter. This kind of cistern is one of the structure made to form Golgi complex. The second structure is tubules. These tubules either branched or unbranched. And the next one is, third one, vesicles. From the margins of these cisternae, due to pinching means cutting, vesicles are formed. These are spherical shaped structures. And the fourth one is Golgian vacuoles. Golgian vacuoles. So these Golgian vacuoles also spherical shape. These are having materials, filled with materials. Okay. The structure of Golgi apparatus or Golgi complex is made up of 4 different structures. The first one is cisternae, second one is tubules, third one is vesicles, fourth one is Golgian vacuoles. So each Golgi complex is made up of 4 to 8 a flattened sac-like structures is called cisternae. These cisternae arranged concentrically interconnected to each other. Concentric means very close to each other. So these 4 to 8 cisternae arranged concentrically interconnected to each other very near to nucleus. It is darkly stained or highly colored cell organelle. Clear? So, due to concentric arrangement, interconnection of these cisternae, one side towards the nucleus, it forms convex shaped. What is the shape? Convex shaped. And towards outside, it looks like concave shape. Convex shaped and concave shaped. The convex shaped Golgi complex towards nucleus is called as cis phase, is also called as forming phase, is also called as young phase. The cis phase or forming phase or young phase, its shape is convex shape. And opposite to convex shaped, the next one is concave shaped. It is also called as trans phase or mature phase. Clear? There are 2 different phases, sites are present for Golgi apparatus. One is cis phase, another one is trans phase. Cis phase is convex shaped and trans phase is concave shaped. It is also called as youngest phase, it is called mature phase, it is called as a forming phase. And from the margins of these cisternae, there is branched or unbranched structures is called tubules. Due to pinching or cutting of margins of cisternae, it forms vesicles. Now, this is the basic information about the structure of Golgi complex. This Golgi complex is the second endomembrane cell organelle having what kind of functions? So, functions of Golgi apparatus. The first and very most important function of Golgi complex is process and packaging and translocations. process and packaging and translocation phenomenon done by one of the function of cell organelle Golgi complex. Sir, what is process and everything? Sir, maybe you have the doubt. Very simple, amma. See here, the rough endoplasmic reticulum synthesis proteins. These proteins are packed in one vesicles. Protein with vesicle. The smooth endoplasmic reticulum help for synthesis of lipids and steroids. These are also packed in vesicles. Vesicle with lipids and steroidal hormones. These vesicle with protein, vesicles with lipids and steroidal hormones releases from the smooth endoplasmic reticulum and enters into the first phase of Golgi complex is convex phase or convex shaped phase is called as cis phase. The newly formed vesicles attaches to the youngest phase or forming phase or cis phase. Now these vesicles are binded to cisternae at cis phase. These vesicles will move from one cisternae to another cisternae and finally releases at concave shaped phase is also called as trans phase. The newly formed vesicles with the proteins and newly formed vesicles with lipids and steroidal hormones first binded at the cis phase and it's moved from one cisternae to another cisternae and finally released from the trans phase or mature phase. Now these components are modified. For example, if the vesicles are having proteins, it's moved from cis phase to trans phase. Along with these one cisternae to another cisternae, these proteins are modified. These modified proteins we called as glycoproteins. This phenomenon is called as glycosylation. Proteins are modified in Golgi apparatus into glycoproteins. Even these vesicles are having lipids. The lipids are modified into glycolipids by the process of glycosidation. The modified proteins is called glycoproteins by the process of glycosylation. The modified lipids is called glycolipids by the process of glycosidation. The modification of proteins and modification it's happen in which of the following endomembrane cell organelles means Golgi complex. And the process of transfer of these vesicles from cis phase to trans phase and packaging of these vesicles and processing of these vesicles, it's happen in Golgi complex. It is one of the function. Clear? And one more. These Golgi complex, it's transfer these modified proteins, glycoproteins and modified lipids, glycolipids. Inside the cell is called intracellular transportation and outside the cell is called extracellular transportation. These modified proteins and modified lipids based on the requirements, maybe it is transported inside the cell is called intracellular transportation, outside the cell is called extracellular transportation. That is the third function of Golgi complex. First one, second one, third one and intracellular and extracellular transportation, fourth one. For example, the next one is, you know very well, this is the sperm. This is acrosome. In sperm, the hood-like structure is called acrosome. The acrosome in sperm formed by Golgi complex. Clear, ma? And one more important characteristic feature. These Golgi complex generally root cap, you know very well. This is root. This is called as root cap. The root cap rich of Golgi complex. Then these are releases secretions. So whenever the Golgi complex rich in root cap, whenever these are secreted, the root tip easily penetrated inside the soil. For easy penetration of root tip into the soil due to presence of rich of Golgi complex in root cap. These are the common functions of Golgi complex. Clear? And at mature phase, vesicles are formed from the trans phase. These vesicles can form lysosomes. Clear? So the next endomembrane cell organelle is lysosomes. And one more, functionally and structurally, the Golgi complex, it is related with or associated with endoplasmic reticulum. This point is very most important for NEET medical entrance exam. Let's we go to the third endomembrane cell organelle is lysosomes. Okay. The third endomembrane cell organelle is lysosomes. So heading is lysosomes. These are also one of the endomembrane cell organelle. It is also single membrane bound cell organelle. It was first discovered by one of the dynamic scientists. His name is Christian D. Dewey. The scientist Christian D. Dewey, he was first discovered lysosomes. These are the third endomembrane cell organelles. How lysosomes are formed, we discuss. These lysosomes we are also called as polymorphic cell organelle. Why the lysosomes are called as polymorphic cell organelles, we discuss. Which of the following endomembrane cell organelle is called as polymorphic cell organelle? Means lysosomes. Lysosomes is commonly called as suicidal bags. Why these lysosomes is called as suicidal bags? We discuss in this class. In the year of, the scientist Christian D. Dewey, he was first discovered lysosomes. It is the third endomembrane cell organelle. It is a spherical shaped single membrane bound cell organelles. So here, for example, this is a single membrane bound cell organelle. A single membrane bound cell organelle, spherical shaped structure. It is an endomembrane system. It was reported by Christian D. Dewey. These lysosomes are having enzymes. The name of enzymes is called hydrolytic enzymes. Name the type of enzymes present in lysosomes means hydrolytic enzymes. All these enzymes are very active at pH. Acidic pH means less than 7. If the 7 pH is neutral, more than 7 is basic, less than 7 is acidic. The enzymes present in lysosomes is called hydrolytic enzymes. All these enzymes are active at which pH means acidic pH. The type of hydrolytic enzymes present in lysosomes means hydrolases, carbohydrases. Carbohydrases, it is one hydrolytic enzymes. They are responsible for digestion of carbohydrates. Lipases, it is an enzyme responsible for digestion of lipids. Nucleases, it is an enzyme responsible for digestion of nucleic acids. And proteases, it is an enzyme responsible for digestion of proteins. All these are a type of hydrolytic enzymes present in lysosomes. All these enzymes are very active at acidic pH. And one more, this cell organelle was first discovered by Christian D. Dewey. This cell organelle is also called as polymorphic cell organelle. Reason is there are 4 different types of lysosomes are present. The first one is primary lysosomes. The second one is secondary lysosomes. And the third one is residual bodies. And the fourth one is autophagosomes. Clear? Why the lysosome cell organelle is called as polymorphic cell organelle means it has more than 3 forms, such as the primary lysosomes, secondary lysosomes, residual bodies and autophagosomes. More than 3 forms present in lysosomes, then the lysosomes is called as polymorphic cell organelle. Poly means many morphological type of lysosomes. Now, what is primary lysosomes and what is secondary lysosomes? We discuss in this class. Listen carefully, it's very most important. For example, see here. This is a Golgi complex. At trans phase, a mature phase in Golgi complex is called as trans phase. It is shape is concave shape. From the trans phase, a newly formed spherical shaped single membrane bound vacuole or vesicle is called as primary lysosome. Are you understood or not? A newly formed or freshly formed lysosomes at mature phase or trans phase of Golgi complex is called as primary lysosomes. Enough. These primary lysosomes are not having any digestive enzymes. So, digestive enzymes are absent in primary lysosomes. Now, the question is, what is secondary lysosomes? Clear, ma? For example, this is a plasma membrane. This is outside the plasma membrane cell. If the substances, either liquid substances or solid substances, binded to plasma membrane, then plasma membrane of cell will be folded. Then the liquid or solid substances present in the vesicle-like structure of plasma membrane, these are called as phagosomes. Phagosomes. The phagosomes are formed from the plasma membrane of cell. So, if the phagosomes are reacted with the primary lysosomes, then it forms secondary lysosomes. Secondary lysosomes also spherical shaped single membrane bound cell organelles. These secondary lysosomes are having digestive enzymes like proteases help for digestion of proteins, lipases help for digestion of lipids, nucleases help for digestion of nucleic acids, and carbohydrates help for digestion of carbohydrates. All these enzymes are very active at acidic pH. Digestive enzymes are present in primary lysosomes or secondary lysosomes? Secondary lysosomes. These are help for digestion of food. The next, these secondary lysosomes forms tertiary lysosomes is also called as residual bodies. Residual bodies. This is the third type of lysosomes. Listen carefully. So for example, this is also spherical shaped single membrane bound type of lysosome. After digestion of food with the help of digestive enzymes in secondary lysosome, some of the undigested food, all food is not digested, some food is undigested. That undigested food present in lysosomes is called as tertiary lysosomes or residual bodies. So these undigested food with residual body attaches with plasma membrane for sending off these undigested food to out of the cell. To sending the undigested food out of the cell with the help of which of the following type of lysosomes means residual bodies or tertiary lysosomes. But example, in liver cell and in muscle cell, these residual bodies are fails to send off the undigested food out of the cell. Then the undigested food, it is stored in liver cell and muscle cell, and whenever this phenomenon is failed, this is called as effagy. Sending off undigested food with the help of residual body to out of the cell is called effagy. If the effagy is failed, those undigested food, if in case stored in liver cell and muscle cell, those people they are getting arthritis joint, means joint pains, and gout disease. Gout means burning sensation in muscles, and lung fibrosis. Lung fibrosis means formation of a thick mucus in the lungs, that is called as lung fibrosis. Lung fibrosis disease and gout disease and arthritis joint disease is causes due to failure of effagy by residual bodies. Along with, due to failure of these residual bodies sending the undigested food out of the cell, even some genetic disorders also causes. Nearly 20 different genetic diseases also is causes due to failure of effagy. This is primary lysosome, secondary lysosome, residual bodies information. At last, the last and final most type of lysosomes is autophagosomes. For example, this is the cell. In some cells, old organelles are present and dead organelles are present. The old organelles or dead organelles surrounded by many primary lysosomes surrounding to old or dead organelles, and all these cell organelles help for, all these primary lysosomes help for breaking and digestion of these old organelles or dead organelles. So to digest these old organelles or dead organelles, all these primary lysosomes kill their self. Then these are called as suicidal bags, or it is also called as auto means by self, autophagosomes. Phago means eating. Clear? This is an information about the third cell organelle is lysosomes. Clear? And after lysosomes in endomembrane system, the last and final most endomembrane cell organelle is vacuole. Clear? The last and final most endomembrane system cell organelle is vacuole. Now the class is vacuole. For example, this is one eukaryotic cell, example plant cell. This structure we called as vacuole. The spherical shaped single membrane bound cell organelle present in eukaryotes and in prokaryotes is called as vacuole. But in eukaryotes, true membrane is present. This vacuole was first discovered by one of the dynamic scientists. His name is Spellanzani. The scientist Spellanzani, he was first to discover the structure of vacuole. Clear? So this is a spherical shaped structure. It is a single membrane bound cell organelle. The structure of vacuole having a true membrane. This is also called semi-permeable membrane. In animal cell and in plant cell and bacterial cell, this is called as a cell membrane. Cell membrane is also called as semi-permeable membrane. So like cell membrane, the vacuolar membrane also acts as a semi-permeable membrane. This vacuolar membrane is also called as tonoplast or is also called as vacuolar membrane. It is a true membrane. It is act as semi-permeable. Then it can be sent the molecules or ions or any substances from higher concentration to lower concentration, from cytoplasm of cell to inside the vacuole. Then inside the vacuole, the concentrations of substances will increases. The concentration of cell will increases because of vacuolar plasm. The vacuolar plasm is also called as tonoplasm. Vacuolar plasm is also called as tonoplasm. These vacuolar plasm having water and another sap substances, sap like structures, and excretory materials, and materials which is not used in a cell, materials which is not used in a cell. Those substances can stored in the vacuole. All the waste substances can stored in the vacuole. So that's what is called as storehouse of a cell. Vacuole is also called as storehouse of a cell, or is also called as a repository of a cell, or cell repository. Clear? The vacuolar membrane is called tonoplast. The vacuolar plasm is called tonoplasm. In plant cell especially, this is a plant cell. In plant cell, the vacuole can occupy 90% of cell volume. 90% of plant cell volume occupied by which of the following endomembrane cell organelle means vacuole. This is very most important for NEET medical entrance exam. The size of vacuoles is bigger in plant cell. The size of vacuoles in plants is smaller. Based on their functions, vacuoles is classified into based on their functions. Vacuoles are classified into three types. Okay, such as first one is contractile vacuole. Contractile vacuoles. For example, if you see the structure of amoeba, If you see the structure of amoeba, it has a contractile vacuoles. These contractile vacuoles, it is responsible for excretion. That means excretion of waste substances. The second type of vacuole based on their function is food vacuoles. For example, in protistants, you know very well, all unicellular eukaryotes, either euglena or paramecium, due to intake of food material from outside to inside, a vacuoles are formed, is called as food vacuoles. And the last and final most is gas vacuoles. Gas vacuoles is also called as pseudo vacuoles, means false vacuoles. For example, in blue-green algae or cyanobacteria, example nostoc and anabena, there is membraneless vacuole is present. These are not a sap vacuole. It is filled with gas. These vacuoles are called as inclusion bodies. Whenever it is filled with the gas, it is responsible for buoyancy or floating. These are the three different types of vacuoles. This is an information about the fourth endomembrane cell organelle is vacuole. So inside the eukaryotic cell, many cell organelles are present. But out of all the cell organelles, a few cell organelles function is coordinate to each other. Those cell organelles we called as endomembrane cell organelle. That system we called as endomembrane system. According to NCERT, they are given 4 NCERT endomembrane cell organelles: endoplasmic reticulum, Golgi complex, lysosomes, vacuoles. Clear?